So good morning. My name is Thomas Farr-Porch, CEO and co-founder of Thrive Bioscience in the Boston area. And we started Thrive 5 years ago to automate a neglected but critically important area of life sciences, which is cell culture and stem cell culture. Sadly, cell culture is still performed similarly to the way it was 65 years ago, with few tools, even though it's an $18 billion per year market. Experiments lack comparability across labs and across time due to the variability of cells. So our products and our goal is to introduce previously unavailable instruments and software to consistently grow healthy cells and with data. We are introducing a family of products with increasing degrees of automation from 75,000 to 750,000. We're currently in beta tests of the Cell Assist at Stanford Stem Cell Core, Harvard Stem Cell Institute, Massachusetts General Hospital, and others. The instrument will be launched commercially next June at ISSCR, International Society for Stem Cell Research. And we expect to grow to $90 million in revenue in 2023, with a significant amount of revenue from software subscriptions and from disposables. We have a significant portfolio of 46 patent applications, 7 of which have issued, in part due to the fact that these instruments have never been developed before. We have extensive collaborations, including purchase of instruments with the Broad Institute, Harvard Stem Cell Institute, and just now with a major pharmaceutical company. We've raised $20 million to date from angel groups, family offices, largest of which is Estee Lauder family office, individuals. And we'll be raising a $12 million Series B in the beginning of next year to fund the launch of our first 2 products. We have a staff of 12 who focus on the biology, software, building of prototypes, and we use an outside engineering firm for final engineering and manufacturing. My own background, I've started over 11 companies, 4 with multi-billion-dollar exits. My co-founder and chief scientific officer is responsible for many of the innovations and instruments in sequencing. The chairman of the board of directors was president of the largest laboratory products group in the world at Thermo Fisher. We also have the executive director of the Harvard Stem Cell Institute, which is probably our most important application. Cell culture currently is performed in incubators that only control gases and temperature and humidity with a pan of water. In order to look at the cells or run experiments on them, we have to take them across multiple environments, subjecting them to stress, to contamination, and a lack of documentation. Drug Discovery World stated, cells need carefully tuned environments to survive and maintain their in vivo properties, and today's incubators do not include the features necessary to create these environments. Unfortunately, as a result of poor cell culture, experiments are often variable. Scientists spend a lot of time repeating the same experiment until they get the results that they want. And if an experiment works or doesn't work, it's very difficult to go back and look at why. But more frighteningly is the amount of genomic drift, contamination, mislabeling. National Institutes of Health found between 14 and 32% rates of mislabeling, which means that researchers actually think they are looking at a liver cell, but they're looking at a pancreatic cell. And there's variation across labs and within labs. Our disease models are getting farther from the patient, from us. So clearly, good science needs good cells. Thrive Solutions is a family of instruments. They're all based on the same databases, same modules. And starting with the Cell Assist, which has a list price of $75,000, is currently in beta test at Stanford Stem Cell Core and Massachusetts General Hospital, University of Tokyo, and others. It's a benchtop instrument that images a plate, one plate at a time, and increases the intelligence, in a sense, of the incubator. And it lists for $75,000 and is also available on a monthly subscription basis. The auto imager for $225,000 is an automated version of the Cell Assist for 50 plates. It images plates around the clock, so that when a researcher comes in the morning, rather than spending a couple of hours looking at plates, all that work's been done, reports have been generated, workflows have been prioritized, as well as creating a database for research. And the Alpaca for $750,000, we have 3 alpha versions working and being tested, is full walk-away automation. It's in a closed, controlled environment. Hundreds of variables can be controlled by the user. And it uses intelligent image processing to make the most important decisions. The first product, Cell Assist, we call cell metrics in a box. So a researcher, instead of taking a plate of cells and putting them under a microscope and doing this for several hours a day, puts it into the Cell Assist, 1 plate at a time. And in about 5 minutes, we take 1,470 images, build a database, build some reports, analyze the cells, and provide automated documentation. The benefits that our collaborators have found include it fits well within the current workflow. It does a better job at looking at hundreds of thousands of cells in a period of time. It's automatically documenting, and it's providing data that wasn't available before. Here are a couple of screenshots. So for example, on the left, you'll notice that there are some plates that are growing at a faster rate. They're just outside the oval. So we noticed that, and we asked the beta site what was going on there. Perhaps they were trying to optimize the process to get them to grow closer. But even though it was at a very well-known institute, those 2 plates were mislabeled. And on the right is an example of what we provide for workflow. The researchers set a threshold for differentiation. They wanted to be directed to those wells in those plates where differentiation was occurring, which is what they wanted to clean. So it allowed them to focus on the most important things first. Some of our other beta test sites have found that they've been able to achieve yields in growing iPSCs that nobody else in the world has achieved, and they've been able to reduce screening times on patient cancer cell lines from 18 weeks to 2 weeks by studying the morphology of the cells. Here's a screenshot from the Stanford stem cell core. They noticed that there was a series of cell lines that were not growing particularly well. And it occurred to them that those cells, when they grow, they clump around the outer edge of the well. Well, most researchers don't look at the outer edge of the well, partly because there's a meniscus which makes it more difficult to see. In fact, this cell line was growing at 50% higher rate on the edges than in the inside. And the overgrowth on the outside was affecting the rest of the cell line. The Alpaca we call cell culture in a box. And what's different about it from other automation is its flexibility. The user controls hundreds of variables, and it's combined with image processing. So all that data that's been collected on the Cell Assist develops algorithms for passaging, for dissociation, for trituration, for directed differentiation, for expansion. Cells can remain in there for months at a time. So why would researchers pay $75,000 to $750,000 for these instruments? Well, besides leveraging their time by a factor of 3 to 5, the payback just from saving time in looking at cells is less than 18 months. And the closed controlled environment reduces contamination, cell stress. The automated protocols allow the lab to perform the same experiment each time, combined with automated imaging, which is driving the process, so it removes human judgment. Cell culture is an $18 billion per year market. 3/4 of it is commercial, mostly biotech, pharma, and industrial and cosmetics. And it's growing rapidly at about 10% per year. And its main applications are drug discovery, in vitro toxicology studies, assay development, drug screening. So hopefully this video will show, but I don't have a cursor to, is there any way that you can start that? It is embedded in it, but So, okay. So this is the beta version of the Cell Assist. We expect to sell about 6 of those this year and launch next year. Everything's barcoded, which users absolutely love. They even barcode the incubator it came from, the reagents that are used. So we have 4x, 10x, 20x bright field and phase contrast. We'll add fluorescence in a later model. So this shows you some examples of how you can drill down on data. On those graphs, you can hover over any of the points and it'll show you the data and the images behind it. And since we take 1,475 megapixel images, there's tremendous resolution. You can then run videos, but this one shows you can go back and look at whether a colony was monoclonal or polyclonal. Here the user is grading their iPSCs, their colonies, based on size. So just show you a little bit of the alpaca. Everything comes through an airlock. There's refrigeration, warming for reagents. Each plate is in a cell hotel that's isolated. It uses pipettes, scrapers in a way so it reduces contamination. You can't override it. It's the same imaging as is in the Cell Assist. So the database that you are starting now, when we deliver the alpaca in 2 years, will be useful. So our near-term milestones for the next couple of months are we're adding 5 beta sites and shipping 8 to 10 Cell Assist instruments. Our milestones for next year are the launch of the Cell Assist and sales of early access versions of the automated version. Essentially, we're selling a new instrument each year. The Cell Assist is in beta. The auto imager is in prototype, and the alpaca is in alpha versions. So we've raised $21 million to date in a Series Seed and in a Series A. And we're looking to raise $12 million at the beginning of this year. About a third of it is for the launch of the Cell Assist. About a third of it is for completion of the automated version and launch. And about a third is to complete the re-engineering of the alpaca. And I mentioned our investors include 7 angel groups, such as Life Science Angels, Sidecar Angels, New York Angels, family offices, individuals such as a former CEO of Novartis, and some strategics, pharmaceutical companies just made an investment. The best statements come from customers. As one customer said, "We have a complete lack of tools to help us in this increasingly important task of cell culture. Cell culture may be the only remaining island of outdated manual processes surrounded by an ocean of automation. Help us by bringing automation, analytics, and better biology to life sciences."